leukocyte development, kinetics, and functions. So leukocytes, by definition, they are white blood cells. So white blood cells are also called leukocytes. They are the immune cells responsible for defending the body against infections, foreign substances, and abnormal cells. They circulate in the bloodstream and lymphatic system and can migrate to tissues where they are needed. Leukocytes are also divided into two major groups, and this is based on their appearance under a microscope and the presence or absence of cytoplasmic granules. So first up, we have granulocytes. Granulocytes have visible granules in their cytoplasm and typically possess a multi-lobed nucleus. Eosinophils are an example of this. These cells contain large reddish-orange granules. They are involved mainly in allergic reactions and help defend against parasitic infections. Basophils are another example of granulocytes. They have large dark blue or purple granules. They participate in inflammatory responses by releasing histamine and other chemicals. And then we have segmented polymorphonuclear neutrophils, also known as neutrophils or PMNs. So these are the most abundant white blood cell found in the body, typically. They have a segmented multi-lobed nucleus and are players in the acute inflammation and fighting bacterial infections. And then we have our mononuclear cells. So the mononuclear cells have a single large nucleus without visible granules. Monocytes are an example of a mononuclear cell. These are the largest white blood cell found in circulating blood typically. Monocytes will circulate in the blood and, upon migrating into tissues, mature into macrophages and dendritic cells. They are phagocytic and help in both innate and adaptive immune responses. And then we have lymphocytes. These are smaller cells with a large round nucleus and a thin rim of cytoplasm. They are central to adaptive immunity and include T cells, B cells, and natural killer or NK cells. So the immunity or immune system, we have innate and specific immunity. So innate is non-specific immunity. This is the first line of defense against pathogens. It will act quickly and target a broad range of invaders without any specificity. Phagocytosis by neutrophil is a mechanism. So this is where neutrophils engulf and destroy virtually any foreign particle or microorganism they encounter. Innate immunity includes physical barriers like skin, chemical barriers, and immune cells like neutrophils and macrophages. And then we have specific or adaptive immunity. This immune response is targeted and specific to particular pathogens or antigens. It develops more slowly but provides long-lasting protection and memory. So antibodies produced by B lymphocytes bind to specific targets, also known as antigens, on the pathogens. The adaptive immunity involves specialized cells such as B cells, T cells, and the production of antibodies to neutralize or destroy specific invaders. All right, so neutrophils, which are a type of granulocyte, develop in the bone marrow through a series of well-defined stages. This process ensures a steady supply of mature neutrophils that are ready to respond to infections. So first we have the stem cell pool. This contains hematopoietic stem cells, which are capable of self-renewal or making more stem cells and differentiation into various blood cell lineages. These stem cells serve as the origin for all blood cells, including neutrophils. Then we have the proliferation pool, which consists of cells actively dividing and maturing. This includes several stages in the following order of maturation. So we have common myeloid progenitors. These are multipotent progenitors that give rise to several lineages, including granulocytes. Then we have colony-forming units, which are granulocyte, erythrocyte, monocyte, and megakaryocyte. Sometimes you'll see this abbreviated as CFU-GEMM. These are early progenitors with potential to develop into granulocytes or other myeloid cells. Then we have granulocytic monocyte progenitors. These are more lineage-restricted cells that are committed to either a granulocyte and or a monocyte lineage. Then we have myeloblasts. This is the earliest recognizable granulocyte precursor with a large nucleus and a scant or little cytoplasm. Then we have a promyelocyte. These are cells that begin to develop cytoplasmic granules. And then we have myelocytes. These cells show further cytoplasmic maturation, nucleus starts to condense, and we'll go into these in more detail later. So just FYI, this is kind of an overview right now. And then we have the maturation or the storage pool. This contains cells undergoing nuclear maturation and final differentiation. These cells form the marrow reserve, which is a storage pool ready to release into the bloodstream during times of increased demand. This can include metamyelocytes. This is where the nucleus becomes indented and cytoplasm is more granular. A band neutrophil, which is a nucleus elongated and curved. It kind of looks like a band or a horseshoe. It's still considered immature, but it is close to being fully mature. And then we have our segmented neutrophils, which are fully mature neutrophils with segmented multi-lobed nucleus, which are ready for circulation. So just a quick summary of our neutrophil development is we have that stem cell pool, which is the self-renewal and differentiation stage. We have the proliferation pool, which are where cells are actively dividing through those stages. And then we have our maturation storage pool, which is where there is nuclear maturation in the metamyelocytes, band neutrophils, and segmented neutrophils that are going to be ready for release into the bloodstream. Again, this is what we just talked about. This is just a quick overview of it. All right, so the first one we have is a myeloblast. By definition, the myeloblast is the earliest recognizable precursor cell in the granulocytic or neutrophil lineage. It is found in the bone marrow during normal granulopoiesis. Nuclear features for this, so the shape, it's going to be large. So generally, the more immature or young the cell is, the larger it's going to be. So a myeloblast is large and it's round to oval nucleus shaped. The chromatin is fine, delicate, and evenly distributed. It kind of appears open or just kind of looks immature. There are usually 2 to 5 prominent nucleoli that are visible within the nucleus. And as you can see on that picture there, there's 2 little circles kind of in the middle, kind of looks like eyeballs. Those are the nucleoli. So when you're looking under the microscope, be sure to be looking for those. Generally, they're pretty prominent and you can see them pretty well. Then we have our nuclear cytoplasmic ratio. This is high, which means the nucleus occupies most of the cell's volume. For cytoplasmic features, the color, it's a basophilic blue cytoplasm, and that's due to the abundant RNA and ribosomes still present. There is a moderate amount of cytoplasm compared to the nucleus size. So you can see in the picture that purple circle, big circle, that is your nucleus, and that little bit of blue lining around it is actually your cytoplasm. The texture, so for the cytoplasm, it's going to be smooth, doesn't really have any granules at this stage, and if it does, it's very, very few granules. If it does have granules, it may have the primary or azerophilic granules, and like I mentioned, these are few or absent in the myeloblast stage. But when they are present, they appear as a small kind of reddish purple granule, and it will be found within the cytoplasm. Specific or secondary granules are not present in the myeloblast stage. They will develop later when they become promyelocytes and beyond. So an incidence on finding these, so the myeloblast is a rare cell in normal bone marrow. It represents a small percentage, about 1 to 5% of the nucleated cells found in there. It's not found in peripheral blood under normal conditions for the patients, and increased numbers can be seen though in certain pathologies like acute myeloid leukemia. Then we move on to the promyelocyte. So by definition, the promyelocyte is the stage following the myeloblast in the granulocyte development. It represents an early immature granulocyte precursor found in the bone marrow. For nuclear features, its shape, it's larger than a myeloblast nucleus. It's round or slightly oval. The chromatin is still relatively fine, but starting to condense slightly compared to that myeloblast. Nucleoli, it'll have about 1 to 3 prominent nucleoli, but it'll start to become a little less distinct than in the myeloblast. So you may see them, you may not see them under the microscope. Nuclear cytoplasmic ratio, it's slightly decreased as the cytoplasm starts to increase and the nucleus starts to get smaller. For cytoplasmic features, the color, it still has that blue tint due to the ribosomal RNA still found there. It does have more abundant cytoplasm than the myeloblast as well, as you can tell in the picture. It's starting to get a little bigger around that large purple circle, which is your nucleus. For texture, the cytoplasm will begin to show some granularity as some granules do start to develop. So for those granules, we have the primary granules. Those are prominent, large, dark purple or red granules that are scattered throughout cytoplasm. These are primary lysosomal granules that contain enzymes. Then we have our secondary granules. These are usually absent or they're beginning to appear later stage in the promyelocyte lifespan. There we go. Develop further in subsequent stages. So the next stage you might start to see some of these secondary. The incidence, we have promyelocytes, which represent a small proportion of bone marrow cells. It is more abundant than the myeloblast. It is not normally present in peripheral blood under normal circumstances, but it can be seen in certain hematologic disorders. The myelocyte is the next one in line. So by definition, the myelocyte is a later stage in granulocyte development following the promyelocyte. It is the first stage where specific granules are going to start become more prominent. For nuclear features, its shape, it's round or slightly oval nucleus. The chromatin starts to become more condensed and clumped compared to the promyelocyte stage, and that just helps to indicate that it is maturing. Nucleoli is usually not visible, and if they are, they're very faint at this stage. For the nuclear to cytoplasmic ratio, it is further decreased as that cytoplasm volume increases. Cytoplasmic features, so color, it starts to become less basophilic than earlier stages. The cytoplasm becomes more neutral or pale pink as that RNA content starts to decrease. So the cytoplasm is more abundant, as mentioned earlier, and it's more granular due to the presence of those specific granules. So the primary granules are still present, but they are less prominent than in the promyelocytes. And then we have secondary granules, which are abundant, smaller, and pinkish, or like a neutral kind of colored granule. These granules define the granulocyte type, so neutrophilic, eosinophilic, or basophilic. Myelocytes are found in the bone marrow, but not normally in peripheral blood. These represent a larger proportion of marrow cells during active granulopoiesis. It can be increased in peripheral blood for certain pathological conditions, which we'll talk about later. Then we move on to the metamyelocyte. So by definition, the metamyelocyte is the stage following the myelocyte in the granulocyte maturation. It is an immature granulocyte that shows further nuclear and cytoplasmic development. So for nuclear features, its shape, it is kidney-shaped or indented nucleus. It is not round anymore. For the chromatin, it is more condensed and clumped than in the myelocyte, which indicates that it is further maturing. Nucleoli are absent at this stage, and the nuclear to cytoplasmic ratio is also decreased compared to the prior one. For cytoplasmic features, the cytoplasm is less basophilic, and it's often a pale pink or a neutral color by this point. It is more abundant for the cytoplasm compared to earlier stages as well. The cytoplasm is granular, and that's due to the specific granules. So primary granules are present, but there are fewer than even the earlier stages. And then we have our secondary granules. These are numerous. They also, like before, define the granulocyte type, whether it's going to be neutrophilic, eosinophilic, or basophilic. The granules are smaller and more numerous than in earlier stages. Metamyelocytes are present in the bone marrow as part of the maturation pool, and they are normally not found in peripheral blood, although you may see a couple here and there, in normal patients as well. Appearance in peripheral blood can indicate a left shift or increased marrow response to an infection or an inflammation. Then we move on to a band or a band neutrophil. So by definition, the band neutrophil is an immature form of a neutrophil, which is one stage away from being fully mature, which is the segmented neutrophil. It is part of the granulocytic maturation process in the bone marrow and can be released into peripheral blood during increased demands. For nuclear features, the shape is elongated, it's curved or a band shape with no segmentation. For the chromatin, it is condensed and clumped and more mature than in the metamyelocytes. Nucleoli are absent. They did not come back. We have the nuclear cytoplasmic ratio. Once again, it's lower than the earlier stages. For cytoplasmic features, the color is usually a light pink or pale blue cytoplasm. We have an abundant cytoplasm compared to earlier precursors as that nucleus kind of gets a little more smaller and condensed. And the cytoplasm is also granular due to those specific granules. So once again, the primary granules are present, but there's just not as many than before. And then our secondary granules are numerous and once again well developed, giving cytoplasm a granular appearance. And once again, that's how you get the characteristic of a neutrophil based on these granules, whether it's an eosinophil, basophil, or neutrophil. So incidence, band neutrophils are found in bone marrow and peripheral blood. Normally, they make up 3 to 5% of circulating neutrophils. Their increased numbers in blood often indicate an active immune response, such as an infection or inflammation, and we would call this a left shift. In reporting, so in a CBC or a complete blood count with differential, band neutrophils are reported as a percentage of total neutrophils. Depending on where you work, some places will have you separate them out. It just kind of depends, so make sure you know your place's policies. And then we finally get to that segmented polymorphonuclear neutrophil. So by definition, that is a segmented neutrophil, and these are the mature form of a neutrophil. They are the primary cells involved in acute inflammation and bacterial defense. For the nuclear features, so the shape of the nucleus is it's segmented into 2 or 5 distinct lobes connected by thin chromatin strands. The chromatin is highly condensed and clumped, which indicates full maturation. There are no nucleoli present, and the nuclear to cytoplasmic ratio is low. So cytoplasmic features, color is pale pink to kind of a neutral kind of color. It is abundant, and it surrounds the nucleus. Texture, it'll have granular due to the presence of those granules. Again, primary granules, it'll have small numbers in there, and it contains enzymes that are important for microbial killing. And then we also have secondary granules, and it's numerous and smaller than the primary granules. It contains enzymes and proteins critical for neutrophil function, like lactoferrin and collagenase. Incidence, so we have segmented neutrophils are the most abundant type of white blood cell in peripheral blood in normal healthy people, and it accounts usually for about 50 to 70% of the total white blood cells that are counted. They circulate in the blood and migrate rapidly to sites of infection or tissue injury. If there's an elevated or decreased count, it can indicate infection, inflammation, bone marrow disorders, or other clinical conditions. So neutrophil functions. It plays a role in the innate immune system. So just as an overview, the innate immune system provides a first line of defense against those foreign organisms. It is not antigen specific, meaning it does not target specific pathogens. It offers no protection against re-exposure. The response is the same each time the pathogen is encountered. This will rely on physical and chemical barriers and cellular components to prevent and eliminate infections. Barriers and components, so we have physical barriers such as skin that'll act as a tough protective shield against entry of microbes. We have mucous membranes also that line the respiratory, gastrointestinal, and genitourinary tracts, which trap pathogens. We have cellular barriers, so we have phagocytes, like neutrophils, and monocytes and macrophages, which engulf and destroy invading microbes. For phagocytosis, it's a mechanism where phagocytes engulf and digest pathogens. It involves recognition, ingestion, and destruction of foreign organisms. Now, as far as the neutrophil goes, we have a neutrophil recruitment. So neutrophils are rapidly recruited to sites of infection or tissue damage. Recruitment is driven by chemotactic agents that guide neutrophils to the target. Those chemotactic agents are substances that attract neutrophils and other immune cells by binding to specific receptors on their surfaces. They are produced by microbes themselves or by the damaged tissue as a signal for immune response. Examples can include things like bacterial products, complement fragments, and cytokines. For motility, neutrophils move toward the site of infection using amoeboid motion. This allows them to squeeze through tissues. This motility is essential for their rapid arrival and effectiveness in immune defense. Eosinophils develop from the common myeloid progenitor. The common myeloid progenitor differentiates into granulocyte, monocyte progenitors, which then commit to the eosinophil lineage. Maturation occurs in the bone marrow before those eosinophils enter circulation. For the nuclear shape, this nucleus of the eosinophils is bilobed, which means there are 2 lobes that are connected by a thin chromatin strand. The nuclear chromatin is more condensed than in earlier precursor stages. For the granules, we have those primary granules. They're present, but they're less prominent than in the neutrophils. They contain enzymes and proteins that are important in the eosinophil function. And then we have the secondary granules. These are larger and more numerous than the primary granules. And these are the ones that distinctly make it an eosinophil when you see it. It's responsible for the eosinophil's distinctive red-orange staining with that eosin dye. As far as contents, the primary granules contain the Carlot-Leyden crystal protein. It also contains enzymes like acid hydrolases. Secondary granules contain major basic protein, eosinophil cationic protein, eosinophil peroxidase, and eosinophil-derived neurotoxin. These proteins play key roles in the defense against parasites and in the allergic inflammation. Eosinophils constitute about 1 to 3% of the circulating white blood cells under normal conditions. And as far as tissues go, eosinophils are also present in about 1 to 3% proportions within the bone marrow where they develop as well. As far as eosinophil function, so they play a role in the immune regulation. So they contribute to the immune regulation by promoting the proliferation of effector T cells. They help modulate immune responses, particularly in type 2 immunity, which is important in defense against parasites and in allergic reactions. As mentioned earlier, these are increased in parasitic infections. So during parasitic infections, especially those caused by the helminths or the worms, they are definitely increased. They attack parasites by releasing toxic granule proteins that can damage or kill the parasite. For allergic reactions, eosinophils play a role in allergic reactions like asthma, hay fever, and eczema. They contribute to tissue inflammation and damage through the release of granule proteins and cytokines. Their activation can lead to symptoms that are associated with allergies. So that can lead to eosinophilia, which refers to an increased eosinophil count in the blood. It is commonly seen in parasitic infections, allergic conditions, and some autoimmune diseases, and also some certain cancers as well. Persistent eosinophilia may cause tissue damage due to that excessive inflammation, all the time. Basophils. So basophils develop from progenitor cells located in the bone marrow in the spleen. Their differentiation is once again influenced by several cytokines, notably the interleukin-3 and thymic stromal lymphopoietin. These cytokines promote basophil maturation and activation. For the nuclear shape, basophils have once again a bilobed or S-shaped nucleus. The nucleus is often obscured by these cytoplasmic granules because they are so dense, but generally you can still identify it if you need to. Chromatin is condensed, indicating that it is mature. For granules, basophils contain numerous large dark purple or basophilic cytoplasmic granules. These granules store inflammatory mediators such as histamine, heparin, and proteoglycans. Granules are involved in the allergic responses and inflammation. For concentration, so in the peripheral blood, basophils are going to constitute about 0 to 2% of circulating white blood cells. And in the tissues, basophils are less common than even in the blood, and it's generally less than 1% of the leukocytes found in tissues. So mast cells, by definition, are not leukocytes, but they are tissue-resident immune cells. They play key roles in immune responses, especially in allergic and inflammatory reactions. For cell development, the mast cells originate from mast cell progenitors, which are derived from progenitor cells in the bone marrow and the spleen. These progenitors circulate in the blood and complete their maturation only after migrating to tissues. Maturation of mast cells depends on the local tissue microenvironment where they reside, which will help influence their function and phenotype. For functions, they have effector cells in allergic reactions. So mast cells release histamine and other mediators upon activation by allergens, which can lead to symptoms like itching, swelling, and bronchoconstriction. It plays a role in inflammatory reactions as well, so they contribute to inflammation by releasing cytokines, proteases, and other substances that can recruit and activate other immune cells. So mononuclear cells. First one up is monocytes. For monocytes, they make up approximately 2 to 11% of the total white blood cells in peripheral blood. For the absolute number, the absolute count of monocytes in the blood is about 1.3. Development, so monocytes develop through several different morphological stages in the bone marrow. It starts as a monoblast, which is the earliest identifiable precursor of a monocyte. It is rarely seen in normal bone marrow. And morphologically, this is similar to a myeloblast, which makes them very difficult to tell apart. Then we have promonocytes. This is the intermediate stage between monoblasts and monocytes. It undergoes cytoplasmic and nuclear changes in this stage. And then we finally get to monocytes, which is the mature cell that enters the peripheral blood and eventually migrates to tissues. For nuclear features of a monocyte, the nucleus is large and indented, or kind of even a kidney shaped sometimes. Chromatin is less condensed than of the mature monocytes, appearing more open and fine. There may be some nucleoli present, but they're not always there or prominent. You're not able to see them. For cytoplasmic features, cytoplasm is abundant and basophilic, so that means it's kind of bluish. So like I mentioned, it is usually a blue, maybe a gray blue in color with a kind of a ground glass looking appearance. And the cytoplasm may show vacuolations or small clear places, or it looks like holes. That's kind of a good indicator that you're looking at a monocyte. For granular content, it contains the primary granules, which are lysosomes with enzymes for digestion of phagocytized material. The granules are fewer and less prominent than in early myeloid cells. These granules contain enzymes such as myeloperoxidase, acid hydrolysases, and other degradative enzymes. For nuclear features for monocytes, so the nucleus is large, often kidney-shaped or horseshoe-shaped. The chromatin is more condensed than in promonocytes, but it's less dense than the lymphocytes. The nuclear outline is irregular but smooth, and it does not have any prominent nucleoli. For cytoplasmic features, cytoplasm is abundant in gray-blue with a ground glass appearance. It contains the primary granules that are small and scattered around. Cytoplasm may exhibit those vacuoles, which are those clear spaces, especially when they are activated. As mentioned earlier, it makes up about 2 to 11% of the total white blood cells found in peripheral blood. For maturity, monocytes are mature circulating cells which are derived from promonocytes in the bone marrow. Once in tissues, monocytes differentiate into macrophages or dendritic cells, which help play an essential role in phagocytosis and antigen presentation. For monocyte functions, they play a role in innate immunity. Monocytes participate in innate immunity response by producing nitric oxide. Nitric oxide is cytotoxic and helps kill invading viruses and bacteria. This will contribute to the immediate and nonspecific defense against those pathogens. Also plays a role in your adaptive immunity. So monocytes interact closely with the B and T lymphocytes. They act as antigen-presenting cells, which processing and presenting antigens to lymphocytes to initiate and regulate adaptive immune responses. This bridges the innate and adaptive immunity. They also play a role in housekeeping. So monocytes play that critical role in removing debris and dead cells at sites of infection or tissue injury. This cleanup process is essential for tissue repair and maintaining homeostasis. After migrating into tissues, monocytes differentiate into macrophages, which are highly effective at phagocytosis. Then we get to our last one, lymphocytes. Cell size, lymphocytes are small. They're approximately 7 to 10 micrometers in diameter. They are most common in the peripheral blood. So lots of times they kind of will be, about the same size as a red blood cell, maybe a little larger. Sometimes you can have large lymphocytes, though. These are larger than the small lymphocytes at about 12 to 15 micrometers, and they're often going to be called activated or reactive lymphocytes. For nuclear features, the nucleus is round or slightly indented. The chromatin is dense and clumped, which kind of makes it look like a soccer ball or condensed appearance in the small lymphocytes. Nucleoli are generally not visible in resting lymphocytes, but may appear in those activated or reactive ones. The nucleus typically occupies most of the cell volume, especially in small lymphocytes. For cytoplasm, it's scant to moderate in amount, forming that thin outer rim around the nucleus in small lymphocytes. It stains light to dark blue, so basophilic. Generally, those primary granules, that's lacking. They don't usually have them that you can see, although some large granule lymphocytes exist with cytoplasmic granules. Cytoplasm may appear more abundant and pale in those activated lymphocytes. I know a lot of times people have trouble determining if it's a reactive or active lymphocyte versus a monocyte. Lots of times your nucleus is going to be the one that tells you what is what. For lymphocytes, they have different groups and functional categories. So T cells is the first group. It's involved primarily in cell-mediated immunity. It attacks infected or abnormal cells directly, and it can help regulate other immune cells. Next one is your B cells. It's responsible for humoral immunity, and it produces antibodies that target specific antigens. It differentiates into plasma cells that can secrete antibodies. And then we have NK, or natural killer cells. This is part of the innate immune response. It can kill virus infected and tumor cells without prior sensitization. Recognize stress cells in the absence of antibodies as well. Categories based on immune participation. So humoral immunity participation, we have mediated mainly by the B cells, which produces antibodies that neutralize pathogens or mark them for destruction. Cellular immunity participation, so it's mediated primarily by T cells and NK cells that attacks the infected foreign or abnormal cells directly. And then our lymphocyte differences compared to other leukocytes. So lymphocytes are generally smaller than granulocytes, but larger than red blood cells. So if you look at the picture down below, it's close to red blood cells, but they are larger. They also have a large or dense nucleus with that scant cytoplasm. The function specialized for adaptive immune responses. We have the T and B cells, and innate responses for the NK cells. So unlike granulocytes and monocytes, lymphocytes have specific antigen recognition and memory. For granules, most lymphocytes are agranular, which means they do not have visible cytoplasmic granules like the other granulocytes. For lymphocyte development, this occurs in primarily the lymphoid organs, so bone marrow for the B lymphocytes and the thymus for the T lymphocytes. That's a good way to remember that. B for bone marrow and T for thymus. So during this phase, the lymphocytes mature and develop without exposure to antigens. And then we have an antigen-dependent phase. This takes place in secondary lymphoid organs, such as your spleen, lymph nodes, tonsils, or your mucosa-associated lymphoid tissue, which is found in the intestinal wall. Here, lymphocytes encounter antigens. They undergo activation, proliferation, and differentiation. B lymphocytes, also known as memory cells, they are long-lived cells that remember specific antigens. They enable a faster and stronger immune response upon exposure to the same antigen. We also have plasma cells under the B lymphocyte. They are differentiated B cells that produce and secrete antibodies specific to the antigen that it was encountered. They are effectors of the humoral immunity as well. As far as blood percentages in the peripheral blood, B lymphocytes constitute about 3 to 21% of circulating lymphocytes in the peripheral blood. T lymphocytes, they have an involvement with the thymus gland. So the thymus is essential for the T lymphocyte development. Immature T cells mature and undergo selection in the thymus before entering circulation. For T lymphocytes, they constitute about 51 to 88% of circulating lymphocytes in the peripheral blood. And then we move on to NK, or natural killer cells. These are relatively large compared to resting lymphocytes due to an increased amount of cytoplasm. Their cytoplasm contains aerophilic or primary granules, which are peroxidase negative, which is unlike the neutrophil granules. These granules contain cytotoxic molecules used to kill target cells. As far as circulation in peripheral blood, those natural killer cells make up about 4 to 29% of circulating lymphocytes. B lymphocytes, so antibody production, they're part of that as well. So B lymphocytes are the primary cells responsible for producing antibodies. Upon activation, B cells differentiate into plasma cells that secrete specific antibodies targeting pathogens or antigens. We have an optimal CD4 T cell activation. So B cells play a role in activating that CD4 and T helper cells. This interaction is crucial for efficient immune responses, as the CD4 and T cells help orchestrate both humoral and cellular immunity. Cytokine production as well. So the B lymphocytes produce cytokines that help regulate the functions of various T cell subsets, antigen-presenting cells like the dendritic cells, and macrophages. These cytokines modulate immune responses, influencing inflammation, cell proliferation, and differentiation. So T lymphocytes and NK cells, so that CD4 T helper cell we were talking about, the function is to send signals that activate the immune system. They help activate other immune cells, including B cells, CD8, T cells, and macrophages. This is crucial for coordinating that immune response as well. We have the CD8 and T cells, or cytotoxic T cells. These are responsible for killing cells infected with viruses or intracellular bacteria. They recognize and destroy infected or abnormal cells by inducing apoptosis. Then we have the Treg cells, or the regulatory T cells. These function to regulate or suppress immune system activity. They help prevent autoimmune diseases by controlling excessive immune responses and maintain immune tolerance to our self-antigens. Then we have the NK lymphocytes, or the natural killer cells. These are part of the innate immune system. They kill certain tumor cells and virus-infected cells without prior sensitization, and they provide a rapid response to infected or transformed cells.